Hello everyone. In today's video, I'll show you how to extract bacteriophage DNA for genomic sequencing using commercial DNA isolation kit. Before we begin, here are the materials needed for the extraction. To obtain high-quality phage DNA, your phage lysate should have a titer of at least 10 to 8 PFU or higher. Let's get started. First, centrifuge 5 ml of phage lysate at 13,000 RPM. Always remember to prepare an extra tube for proper balancing. After centrifugation, gently remove the supernatant without disturbing the pellets or the walls of the Eppendorf tube. Next, resuspend the pellet in 1 ml of fresh phage lysate. Mix gently by pipetting until the pellet is fully resuspended. Add 500 microliter of lysis buffer B to the phage lysate and mix using a pipette. Then, add 4 microliter of proteinase K. Incubate the mixtures at 37 degrees Celsius for 24 hours. This step allows proteinase K to digest phage capsid proteins and host-derived proteins, improving DNA release and overall yield. After 24 hours of incubation, add 600 microliter of isopropanol to the lysate and mix by inversion or vortex. Assemble the spin column and collection tube for DNA binding step. Add 600 microliter of phage lysate to the column and centrifuge according to the kit instructions. Carefully lift the column and discard the flow-through. Repeat this step until all phage lysate has passed through the column. Wash the column 3 times using 400 microliter of wash solution to remove residual proteins, salt, and other contaminants. Centrifuge the column for 2 minutes to completely dry the resin. At this stage, the phage DNA should be fully bound to the column. Discard the collection tube and place the column into a new, clean Eppendorf tube. Add 30 microliters of nuclease-free water directly onto the column membrane, then centrifuge for 1 minute to elute the DNA. Once elution is complete, remove the column. Your phage DNA is now ready for whole genome sequencing. You may also store the DNA at -20 degrees Celsius.